Good afternoon. My name is Bruce Walker with the Reagan Institute of MGH, MIT, and Harvard. I'm quite honored to moderate this first ever virtual event for Keystone Symposia. As many of you probably know, Keystone Symposia convenes some of the leading life science research conferences worldwide. This particular webcast is part of their effort to bring more science to more people. And along those lines, I'd like to welcome online participants from more than 80 countries who are participating today. I'd also like to thank Gilead Sciences for sponsoring the webcast. And I'd like to tell the audience a couple of things to help you engage during this panel discussion. There's an Ask Question button in your browser window, and in order to ask a question, all you need to do is click on that and send a message to one of the panelists to be able to address during the period we're speaking here. If we don't get to your questions today, we obviously can't get to all of them. I urge you to join Keystone Symposia for their joint HIV Vaccines and Pathogenesis conferences in March 2014 in Banff, Canada, where there will be a specific workshop focused on this same topic of HIV/AIDS strategies for an endgame. Now to introduce our panelists. Mike Cohen is an MD and Director of the Division of Infectious Disease and the UNC Institute for Global Health and Infectious Disease at the University of North Carolina at Chapel Hill. He's the co-PI of the HIV Prevention Trials Network and a clinical infectious disease specialist. Welcome, Mike. Thanks for being here. Next is Betsy Herold, also an MD, Professor and Vice Chair for Research Development in the Department of Pediatrics at Albert Einstein College of Medicine in New York, and the Director of the Translational Prevention Research Center at Einstein, where she's leading the development of a vaginal ring for HIV prevention. Welcome, Betsy.
Thank you.
Julie McElrath, MD, PhD, is the Senior Vice President and the Director of the Vaccine and Infectious Disease Division of the Fred Hutchinson Cancer Research Center in Seattle. She also leads the Seattle HIV Vaccine Trials Network and is a clinical specialist in infectious diseases at the University of Washington. Welcome also to you, Julie. And finally, Gary Nabel, MD, PhD, is the former head of the Vaccine Research Center at the National Institutes of Health in Washington, D.C., where he oversaw vaccine development efforts until 2012. He's now Senior Vice President, Chief Scientific Officer, and Deputy to the President for Global R&D at Sanofi. Welcome, Gary.
Thanks, Bruce.
So let's go ahead and begin. And let me make a few comments first regarding where we stand right now. There have been more than 35 million people infected thus far by this epidemic and more than 36 million deaths. We're 30 years into trying to find a solution to this epidemic, and yet there's no clear solution that has been forthcoming. Despite that, I think everybody on this panel would agree that there's a sense of increasing momentum that has been generated within the field. There have also been some clear advances in prevention from PrEP trials and also data from animal models and one human study suggesting that effective HIV vaccines might indeed be possible. In that setting, we're now facing constrained budgets, and I'd like to begin this discussion by asking a few questions of the panelists and then come back to focus on four distinct areas. Those areas are test and treat, pre-exposure prophylaxis, HIV vaccines, and the potential for HIV cure. So for the first question to the panelists, from the goal of ending the HIV epidemic, what do you see as the most exciting recent developments to guide strategies for an endgame? Mike, maybe you can begin.
I think that, as you pointed out, the trajectory of the epidemic has been downward, in some places dramatically downward. And I think that's not an accident. That's ascribed to combination prevention strategies that are having a big impact. People have changed their behaviors as a result of all the information and education that's provided. And then I think more recently, as we've rolled out antiretroviral therapy to more and more people, we're seeing that treated people are rendered much less contagious. And that has an immediate effect on partners and a population level effect. A recent study published in Science, we might talk about later, suggests a fairly dramatic decrease in incident cases of HIV when a relatively modest number of people are efficiently treated or successfully treated. So I think that's quite an exciting advance.
Betsy, do you want to make some initial comments?
Sure. I think that, as Mike pointed out, we've had a lot of initial successes. We have a long way to go, and I think all of us would agree that the ultimate eradication will probably come with vaccines. But I think in the short term, we have some real potential out there in the form of pre-exposure prophylaxis, or PrEP as you called it, which can come either as a systemically administered drug or a vaginally or topically delivered drug, which can help protect people. And even if it's only partially protective, it's going to have a major impact, particularly in the most vulnerable populations, including, for example, young women between the ages of 15 to 24, who currently really have the highest proportion of new onset disease.
And Julie, what excites you in terms of recent developments?
So I'm most excited about some recent developments in the vaccines for HIV. And in this case, it's always been considered that the ability to induce broadly neutralizing antibodies would be a really cornerstone activity for vaccines to induce. And recent studies now have shown that individuals with HIV infection can develop these about 25% of the time, and more studies of these particular broad neutralizing antibodies have now yielded the recognition sites, and these epitopes can then be looked at more carefully for binding sites and then modeled structurally for vaccines in the future. So both the ability to use these antibodies potentially as immunoprophylaxis agents, as well as ways to identify the epitopes that they're recognizing, hold real hope for a vaccine in the future.
And Gary, what excites you?
Well, Bruce, for me, I'm quite enthusiastic about pursuing vaccine research, and I think it's actually helpful sometimes to take a step back and ask, what can vaccines do to help us? I think maybe the most striking example in my mind is the history of what we did with smallpox vaccine, where if you look in the 20th century, the impact of that disease was, like AIDS, devastating. There were 375 million people who died of smallpox in the 20th century alone. That's 10 times more than the number of people that were killed in all of the world wars in the 20th century. And yet, when we learned how to implement the vaccine that we actually had a vaccine, but it hadn't been widely implemented. By the late 70s, we essentially took the caseload down to zero. We eradicated the disease. So as a public health tool, vaccines have unprecedented success. I think for HIV, we're clearly not at the point where we were with smallpox in the 1970s. But as Julie pointed out, I think there have been recently some very encouraging advances. And I think that the knowledge that people can make these broadly neutralizing antibodies, because the ones that Julie spoke about were isolated from HIV-infected individuals, gives us a lot of encouragement that that could be done if we're smart enough to engineer the right kind of vaccine. And our understanding of the structure of the virus and those critical determinants on the surface now give us a tool that we can use going forward to create what we call structure-based vaccines that I hope can get us to the level where we need to be while we're still pursuing some of the more immediate leads that can help contain the epidemic.
Yeah. So it seems like we have there's no question that there are advances being made, and one I might also mention that excites me is the demonstration, at least by proof of principle, from Louis Picker using a CMV HIV vaccine, that you can actually protect animals from productive ongoing infection. And what's remarkable there is that the animals clearly seem to get infected and then clear it, which I think really changes the the way that I think about the prospects for vaccines. But there's no question also there's been tremendous evidence that some of these other prevention methods are able to work right now. So I guess the second question I would ask is, given all these recent advances, how should they guide our immediate action that we take right now? And what's the opportunity cost involved in making one thing a priority versus another thing a priority? So maybe, Mike, do you want to start again on this?
Yeah, I'll stay with the same theme. I think that we spent so much time and money developing antiretroviral drugs that are very effective for treatment. And then in the last couple of years, it's been clear that the same drugs can be used to prevent HIV infection in a couple of different ways. Treating infected people renders them less contagious. The current guidelines worldwide call for 26 million people to be treated immediately, and what we really need to do is treat everyone to get the maximal coverage to see the maximal benefit. If we do that, we won't be working around the margins. So the research issue is, how do we do that efficiently? How do we make this available? How do we identify infected people and make these drugs available in a way that is cost-effective, most cost-effective, and as quickly as possible? It's very much like the smallpox rollout. I think that rollout took a lot of energy, and it's going to take a lot of energy to utilize the tools we have. The second point, we'll get more into what Betsy and others, and I and others will talk about, is the use of the same drugs for HIV-negative people as pre-exposure prophylaxis. That clearly can play a major role, especially in some special populations. So I would stay with investing in research efforts at deploying tools that are readily available right now. In terms of vaccines, one last point, I'm terrifically excited about vaccines, as is everyone in this panel, but I think the discipline to wait to do late phase clinical trials until we have agents that have sufficient biological plausibility is an important discipline, and that's something we probably need to talk about.
Betsy, do you want to comment about that?
I think Mike brought up two different areas. One is what is referred to the work he and others have done in test and treat, which is rolling out a system whereby we can test people who are at risk for HIV and getting them on therapy. And we know that works for HIV the same way it works for tuberculosis, something we've done for years and years and years. But I think that the second thing we have out there is we have 2 or 3 major studies showing proof of concept that if we take at-risk people who are HIV negative, who come out of, let's say, test and treat because they're at risk and are negative, and we put them on prophylaxis, be that an oral or a systemic approach to prophylaxis, so taking Truvada, for example, as a pill, or we use a vaginal, in the case of women, for example, vaginally delivered product, again, something like tenofovir gel. We have a terrific proof of concept study done in South Africa, the Caprisa 004 study, which clearly shows if women use it and apply the gel in an at-risk situation, they are protected from getting disease. So I think the real challenge moving forward is, how do we address the adherence problem? Are these the best drugs? Are they the right drugs? How do we decide which drugs to move forward? And how do we do this so that we implement it in a safe and effective way?
Mm-hmm. Julie?
Yes. Well, from a vaccine point of view, I believe that with a recent excitement about the broadly neutralizing antibodies, it's going to be very important that envelope immunogens, potentially really subunit proteins, be manufactured on a quick pace so that they can get into humans' phase 1 trials quickly. So the production line needs to really speed up, and we recognize that just knowing how long it's taken to see a signal in the Thai trial, the RV144 trial, to actually being able to move forward with that. So just getting the pipeline moving more quickly. And then the second part of that would really be to be able to conduct smaller phase 1 studies, potentially in parallel with the larger studies, to try to understand in more depth what the vaccines are doing, what types of immune responses they're inducing, and how could one eventually harness those and target them better toward more protective epitopes.
Gary, do you want to make a couple of comments about this?
Well,
sort of how to prioritize where we go right now.
Yeah, you know, from my perspective, I like to try to divide things into sort of two different categories, because I think you tend to approach them qualitatively different. I think on the one side, we've heard a lot about implementation. Given what we know now, how do we implement that knowledge to impact on the epidemic? On the other side, and I think these issues of how do we take this knowledge that it's important to test early and to treat early and get that working in a very complex world where there are lots of people living under different circumstances with different healthcare systems. That's critical to actually having an effect on the epidemic. So I would say the implementation science is clearly an area of opportunity. The other side is the basic science, the research on understanding the natural history of infection, the mechanisms of latency. And there, I think, we have begun to see some great advances, as Julie said, not just in the definition of broadly neutralizing antibodies, but understanding how those antibodies are actually formed. What's the pathway that the immune system takes from early B-cell precursors to a mature antibody-producing cell, understanding that, using rational design of proteins to elicit that. I think we're reaching a new era where we might be able to crack this old scientific problem of once we have a monoclonal antibody, how do we go back and design a vaccine that could elicit that antibody? And so I think those are wonderful opportunities. And the only other point I'd like to say is that on the implementation side, I think we do have, and maybe we can talk more about this, we do have this other age-old issue of, when do we stop studying and start doing? Or what's the right transition point? And I think that's going to be critical to work out in the coming years.
And are we going to resolve that today here?
You'll tell us.
Okay. Well, maybe with that sort of as an introduction, maybe we can go to these four areas that we talked about: test and treat, PrEP, vaccines, and the prospects for an HIV cure. So, in terms of test and treat, I think there's certainly clear evidence that that's something effective. But from my perspective, one of the things that I really worry about with test and treat is that we're testing a lot of people that we're telling in high incidence areas who are telling that they're negative when actually they're positive. We had done a study in South Africa where we took individuals that were testing negative at regular voluntary counseling and testing sites and did HIV RNA on those samples. And in certain populations, up to 1% of those people were actually undergoing acute infection at the time that we saw them. How do we deal with the issue of acute infection when we start talking about test and treat? And maybe, Mike, do you want to put this in a little bit of perspective in terms of how you see this rolling out?
Right. Well, I think again, we have to go back a little bit. So over a period of years, based on observational studies in a randomized controlled trial, we came to believe and conclude it becomes axiomatic that a properly treated person is rendered no longer contagious. So there were at least 11 observational trials where within couples there were no transmission events observed once the index case, the infected person, was being treated. Then in the 052, HB10 052 trial, there was at least a 96% prevention over a couple of years of HIV transmission when you treated the index case. That's the first point. And then as people got excited about that, because they just saw the potential, observational population studies have almost all moved in the same direction. Almost all, because some populations haven't benefited. We can talk about that a little bit later. But for the heterosexual large populations, we then observed that where antiretrovirals were rolled out and used more readily, incident infections in the population became less common, were reduced, sometimes dramatically. Having said that, it brings up the very point you made. As we test people and treat them, for sure we're not going to find everybody. For sure we're going to miss people with acute infection who, as I think you would argue, are potentially maximally contagious. Maximally contagious because of very high viral load, of a single virus that just came from one person and is growing, ready to go to the next person. That virus is probably more contagious for a variety of reasons. So that window is a problem. But having said that, In the absence of doing anything about acute infection, the observational studies suggest that, again, with a modest level of coverage of 40 or 50% of the people who need ART, you start seeing these very dramatic benefits to the population. So I think we should work on acute infection but not be distracted by it. And that's probably my best answer.
Okay. How much coverage do you think we actually need to have in order to really have an impact with test and treat?
No one knows the answer to that. And in fact, that's a great segue to the question being asked in an orderly way. We saw, again, in KwaZulu-Natal, South Africa, that with 40 or 50% coverage, there was, for every 1% increase in coverage, there was like a 2% decrease, almost a 2% decrease in HIV incidence. A very dramatic observation. But 4 clinical trials, large clinical trials, are underway. Some supported by the U.S. government, some supported by the French government, the Gates Foundation. There's one in Botswana, there's one in Uganda, there's one in Zambia and South Africa, and there's one in KwaZulu-Natal following up on the earlier observation. These very large trials are designed to answer your question exactly. Is this hypothesis correct? And to define the amount of coverage necessary to fully realize the benefit of antiviral therapy. The only other thing to add is, it probably is time to stop debating treating people. Okay, the clinical benefits to the individual. We're focused on transmission benefit or prevention. Of course, that's important, but health is preserved by treating people very early. CD4 count goes up. People join the workforce. They live actually completely normal duration of life. So to me, this debate is over. In the United States, of course, our guidelines are for immediate treatment of everyone, including people with acute infection. So I think the coverage is to be, the amount of coverage to be successful is to be defined by very well-designed randomized clinical trials in the field.
Betsy, do you want to add anything to that?
Well, I think that we can move, I think that test and treat and getting people on therapy is clearly going to be hugely beneficial. So I think the next step is, what do you do with those people who come in who are tested and are negative, forgetting the acute infection question for a moment here. And those are the people that really need to be offered some other prevention strategy, because they're by definition telling us they're a high-risk population. And that's where I think for now, until we have an effective vaccine, we need to start to work harder on how do we select, implement, and optimize the use of pre-exposure prophylaxis. So are those people candidates for a prevention oral pill right now, like Truvada? Are they candidates for some kind of topical approach? As we move forward in research, are they going to be candidates for some of the newer types of agents that we're working on? Intravaginal rings, which might overcome some of the problems of adherence. Sustained systemic therapies, drugs like rilpivirine, which is a non-nucleoside reverse transcriptase inhibitor. This is just an example. There are other drugs out there. But that can be given, and it's long-acting, so it might work like the hormone Depo-Provera. You might be able to take a shot once every few months, and that might protect you. Is that a good approach? Which are the right approaches? Can we learn from the vaccine field? Can we use monoclonal antibodies as a preventative strategy, either delivering them topically again or delivering them systemically, again, while we're waiting for the ultimate eradication approach?
And still on the test and treat topic, what are the strategies that need to be used to actually expand testing and to do that more effectively?
I think you're the...
Well, I think that, again, this gets the implementation science idea. The best antivirals in the world aren't going to either improve health or stop transmission unless we identify people with infection, the majority of them, link them to care, and successfully offer treatment in a way that suppresses viremia. So most of the energy, a lot of the energy in the prevention field is going towards research to define the quickest, most effective way to accomplish those goals. Certainly, the notion of testing people, in the United States, of course, we embrace universal testing, but we also have some targeted testing, and worldwide, most countries have done a great job at testing more people. But there's no reason to be tested if you're not going to be linked to care and offered antiretroviral treatment. So this is all part and parcel. This idea of immediate treatment and setting up guidelines that lead to immediate treatment are probably the biggest incentive to be tested. Now, there's probably nothing worse than asking somebody to be tested, going to a clinic, and not being given antiviral therapy. those people are lost to follow-up at an incredible level. And like the tuberculosis metaphor, what you've done is you've sent people out who you know are contagious with no therapy, whereas they could be taking a therapy that's both beneficial to their health and will render them no longer contagious. So again, we have a two-part strategy here. One is to get everyone on the planet on the same page. And second, to make countries aware of the opportunity to improve the health of the people in their country by making these drugs widely available. And then the research part of it, which is critically important, is simplifying all the infrastructure required for this purpose. Simplifying the testing infrastructure, the treatment infrastructure, and so on and so forth. And what we've seen is a movement very quickly away from all kinds of requirements for testing to vastly simplified testing. And that's all these are movements in the right direction. That unequivocally, we're going to come back to vaccine development in segment 4, I believe.
Yeah.
But I think unequivocally, while we're working towards that essential goal, we need to not make the prevention of HIV through ART marginal. That's going to reduce a lot of need on this planet.
And you know, one of the issues that may impact this whole test and treat agenda is the issue of drug resistance. And along those lines, I'm concerned by some recent data that I saw related to adolescents who are, you know, in the same way that adolescents often do, rebelling to some extent and not taking medications. And in a small study that Tariro Makadzange had conducted in Zimbabwe, found that a striking number of adolescents were actually had detectable viremia on therapy. How is that going to affect this whole agenda?
Well, I think that's a problem worldwide, and that's a problem in the United States with adolescents, in the developed world, in the developing world. I mean, this is something we all know that you know, if you're non-adherent, there's a higher risk, or you're intermittently adherent in particular, there's a much higher risk of resistant virus. I mean, you know, the approach to that is the same as the approach to a lot of the other difficult to treat situations with adolescents. You know, it's got to be multi-pronged. It's got to include behavioral interventions. But I don't think that those exceptions to the rule, I think that sometimes we get too worried about resistant virus. I don't think that we should not promote and not move forward prevention strategies, test and treat, topical pre-exposure prophylaxis, systemic pre-exposure prophylaxis, because some small percentage may have a resistant virus. Because the cost-benefit analysis, the risk-benefit analysis is still going to favor moving those products forward. We're not going to identify everyone with test and treat, and we're not going to protect everyone with PrEP. But if we protect close to 50% of people, we're going to have a huge impact on the epidemic.
Well, maybe we can focus in a little bit more on PrEP. What is it in terms of PrEP that clearly works in a real-world situation now, and how do we go about implementing what works?
Well, if we're going to continue with the tuberculosis analogy, then what really works is directly observed therapy. Is that feasible? Probably not. But we know from the clinical trials, if people take a prevention product, it will work very effectively, perhaps as much as 90%. So I think that what you're really getting at is how do we implement it? And are the drugs that we've initially tested the best drugs? And I think one of the suggestions from the field is that being adherent to a daily oral drug, or being adherent to a topical gel product that you have to put in very frequently, or you have to remember to put in every time you're going to have sex, may not be the way to go. And so we need to be more creative in the way that we're going to formulate these products. And that's really where the scientific discovery has to come from. And so our intravaginal rings is an example. So similar to contraceptive rings, something that a woman could apply, might be in place for 30 days, might be in place for 90 days. Don't have to think about it, don't have to touch it, and going to protect you for that whole time period. Is that going to be a better approach? Is a systemic drug like rilpivirine going to be a better approach? Are multi-purpose technologies, would we get better adherence if we combined an antiretroviral that's delivered as a prophylaxis with, let's say, a hormonal contraceptive, so that women may be more likely to be adherent to that product? That's a thought. We've not proven that. We don't know that. But I think those are the kinds of techniques that we need to think about as we move forward. And
in terms of the adherence issues related to PrEP, What are the ways? What are the novel strategies that can be used to actually try and enhance adherence?
Well, I think there's two issues there. One is the clinical trial approach. So how do you improve adherence during a clinical trial so you can separate out whether a drug was effective versus, so whether a drug will work if people use it or whether a drug just doesn't work at all. If you have a product that you test in a clinical trial and the results are negative and you don't get protection, is that because the product didn't work or because women or men were not adherent to the product? So that's one question. I think that one is relatively easier to address. We can use all kinds of different assays. We can use rapid drug levels. We can do drug levels in time so people are given back the results of their drug levels. And that will inform people and help them to become more adherent. You know, there have been other things that have developed, dye staining, if you're talking about vaginal applicators. People talk about rings that have some kind of pH probe and temperature probe in them that can determine if people are actually keeping the ring inside. That's nice for a trial. What you're asking, the real question is, what's going to happen in the real world?
Yeah.
And I think the answer to that, we need a behavior scientist up here to do a better job of answering. I don't think we know the answer to that one.
But it's certainly been an issue with the trials that have been done thus far, showing
Absolutely.
clearly that adherence impacts all of these modalities.
The way around that, of course, the way around that you alluded to a little bit was injectable agents. And there are like 4, and what's good about this is there's a limited number of agents to discuss. It makes it a lot easier. So there are only 4 things on the table right now. One of the things is pretty avant-garde, and that is David Baltimore trying to develop a vector that would deliver, that it would allow the host to make antibodies constitutively over long periods of time in high concentration. He's very excited about that and has gotten a lot of press and published some very exciting articles. So that's an interesting technological advance, and that would change all of immunology. By the way, if that succeeded in HIV, then the whole vaccine field would be drastically altered for the foreseeable future. The second idea is one that Gary Nabel has been a leader of, and that is use of monoclonal antibodies for pre-exposure prophylaxis. The Vaccine Research Center, with tremendous effort, was able to identify cells that can make antibodies, a monoclonal antibody that's broadly neutralizing, and the antibodies they're generating have exciting potential for pre-exposure prophylaxis. The more immediate agents are rilpivirine injectable. Rilpivirine is a well-known antiretroviral, and it's been formulated as an injectable agent that would last several weeks. So a person could receive an injection and potentially be protected from HIV. Of course, there's therapeutic potential of those injections as well. And then the most recent agent is a GSK agent called GSK744, which is an injectable integrase inhibitor, which lasts even longer than rilpivirine. And the integrase inhibitors, of course, are a very interesting class of drugs because they require no metabolism to be very active. So there's an immediate benefit of them. And those 2 agents are getting ready for phase 2 trials right now. And I know there's tremendous excitement in industry around those agents, both for treatment and for prevention. So we have this kind of menu of agents that would take away at least some of the behavioral activity. I mean, we take away the everyday pill idea. People still have to go back to clinic and get a shot. That shot has to be comfortable and non-toxic, and they have to really believe it's to their great advantage. Now, we have B12 injections in vast numbers in the United States and worldwide of people who either need B12 or believe they benefit from B12. So there is a history of some injectable agents being used for long periods of time, but time will tell as these drugs are developed.
Along the lines of sort of other new developments in terms of therapies, Gary, maybe you could comment a little bit on monoclonal antibodies in terms of use in PrEP. That now with the recent work done at the VRC and other places showing that some of these broadly neutralizing antibodies can actually be delivered, at least in a SHIV model, and have quite striking, at least short-term efficacy.
Yeah, there are now, I would say, at least 4 independent reports of monoclonal antibodies given in non-human primate models where they prevent infection. There have been 2 recent papers in Nature showing that they can even be effective in a therapy mode. I would say on top of that, and what I find exciting for the future, is that the whole field of antibody engineering has continued to progress in leaps and bounds, and we have ways where we can extend half-lives of antibody. We have ways where we can engineer those antibodies to make them more potent. If we can make them more potent, we can use lower amounts, or if we use the same amount, it will give them a longer half-life, which means fewer injections over time.
So you see this as potentially a therapy, not just sort of a proof of principle or a way to sort of understand what the contribution of broadly neutralizing antibodies might be to guide a vaccine development, but really is something that might see the light of day in terms of a clinical intervention?
Yeah, I see it as both, actually, Bruce. I think that clearly we can learn a lot by just doing these trials with antibodies. It will tell us whether the antibody really works. We don't know that yet in humans. If it does work, it tells us that that's a great target to go after in a vaccine for a traditional vaccine approach. But even if we're not ever able to develop the vaccine to elicit that antibody, if we can make an antibody that can target, say, 2 epitopes on a virus that can persist for 3 to 6 months, that's got potential to do a lot in terms of preventing the incidence of new infections. And then the third alternative, which Mike mentioned, might be gene delivery, whether it's AAV or some other form of gene delivery. So I think that the antibodies do have a lot to teach us, and as you point out, Bruce, I think I would not rule them out at this point as being a potential treatment on their own. The only other point I would add, which I think is fascinating in terms of your issues regarding behavior, is that, and this is something I've learned as I've moved into the pharma sphere, which is that we in basic research often pay attention very late in the process to the interface with the patient. And that's where we often fail. In the pharma industry, there are numerous examples of great drugs that have made it to market, that have passed, and people don't use them.
Well, and isn't, in fact, don't we have evidence of that even already in the PrEP setting, where coitally administered tenofovir gel worked, and daily tenofovir gel didn't because of adherence issues?
Yeah, and it can be something as simple as a pill being too big. And along the lines of what Mike was just suggesting, another very interesting development in the patient interface is the development of devices where you can now inject pain-free. So you could imagine if you had an injectable, whether it be a drug or an antibody, and you had a device where it didn't hurt to inject, and you did that once every several months, that might be something that would be very tolerable to the average person, and it might change our thinking about how likely we would be to adopt a new therapy. So I think this idea of being more attentive to patient behaviors and using new technology to smooth those interfaces.
Well, let me actually bring in, one of the things that we're able to do on this webcast is actually get questions from the audience, and there's a question that's come in from Derek Riley Stein at the University of Manitoba, and he's wondering if the panel believes that PrEP will hamper the progress of HIV vaccine trials, particularly with respect to minimal standards of care if PrEP is rolled out. And maybe, Julie, do you want to,
Yeah.
I'm sure you've thought about that a lot in the context of the Trials Network.
Yeah, so just as some background, in a recently conducted Phase 2b efficacy study that we've done, and it was reported over the last month in the New England Journal, Scott Hammer, the first author, while the vaccine regimen itself was not efficacious, this was 3 DNAs followed by an Ad5 expressing HIV genes. These individuals in the study were offered PrEP if they wanted to take it, and surprisingly, there was very low uptake, very low percentage of PrEP associated with the vaccine. study. So it really, to me, highlights the fact that if we are going to see these used, and if they could have impact on vaccine studies, then it's likely going to need to be some type of pre-exposure prophylaxis that's going to be easy to use, that is going to be something that eventually they could stop without feeling like there's a major impact. Something that they could, take with sort of fairly low maintenance. And so I think that's one part of it. But then on the other side of that is that I think it is going to be very important that we begin to think more in combination approaches. That, this sort of making sort of making boundaries between, what's a vaccine that's an active immunization versus a passive approach or pre-exposure antiretroviral treatment. These are all going to blend over time, and so we need to really try to understand well, what are the features of each one of these implementations, and be able to sort out, what's the major driving force.
Well, and that actually relates to another question that came in from Steve Wakefield. He says, Betsy says we need to find biomedical interventions to prevent infection. Mike keeps going back to activity after folks are infected. Why isn't someone designing a strategy that will utilize the tools along a continuum? Don't we still have a greater number of persons at risk than are already infected? And maybe Mike, you want to take that one on?
Well, I think it's
or
correct.
Betsy.
I think it's correct. I mean, I think it's not an not an argument. I mean, Betsy indicated that. Well, but I do think it's tailored in a way, and this tailoring is really important. It gets back to something. It's like, okay, not everyone is going to benefit from PrEP. It's going to be a selected group of people at the highest risk who need to be offered that opportunity, just generically, because we know that from when we look at the kind of population level acquisition of HIV, we can see how this goes. But to ignore that ecological information is not right. So there needs to be some targeting of these tools. It gets at something about combinations that you mentioned. I just want to come back to about, it seems to me one obvious coalescence is that the newer broadly neutralizing antibodies, at least the natural immunity, takes years to develop. It seems very likely to me that the immunogens, if we're able to make them, are not going to take a few months. It's not going to be like an influenza vaccine. This is going to be a special kind of vaccine. And so if we did offer that vaccine, it took a year or 2 years for full immunity, you can see how the PrEP field would marry the vaccine field, and that people would participate in a new trial design that would optimize everything we know to protect the highest risk people. And I'm sure a lot of people are thinking about this.
I mean, I think we can borrow from the analogy of what's happening in infected individuals, that it could take 1 to 2 years to sort of get the right sequence of immunogens to finally get to that broadly neutralizing epitope. But it may be that over time we'll get smarter about what these antibodies are actually seeing, so that we may be able to compress that time a little bit more. And then they'll obviously still have a pretty healthy immune system, so maybe that'll be some benefit as well.
Well, and I think we should come back to that again during the vaccine part of this discussion. Another question I think that we have to deal with is that as we're sort of exploring more and more approaches and developing more concepts, a really critical question, given constraints on resources, is what concepts, how many concepts do we actually take forward, and who should decide on what concepts go forward?
So I think that question actually also addresses the question about the impact of PrEP on vaccine trials. So I think there's an issue out here that we need to address. We're going to come to a stage pretty soon where we can't do placebo-controlled trials. If we want to compare one form of PrEP, whether it's oral PrEP or a topical PrEP or a vaccine, the control arm likely has to be offered, as Julie just pointed out, a form of PrEP that already exists. Mm-hmm. That's going to make trial design very difficult and frankly more expensive. And so I think
And how close are we to that?
I think we're very close to that. I mean, I think some people would argue right now if the Capriso 004 trial, which was the vaginal tenofovir gel, is repeated in the FAX trial, and those data may be out as soon as within the next year, or the dapivirine intravaginal ring, which is an ongoing trial, and those data may be out by early 2015. If either of those look look very promising, and that doesn't have to be a home run. That can be partial protection. We're going to have to move forward by comparing the next generation product against that product. And so I think there's going to be a lot of important scientific questions we address. You know, how do we select the next products? How do we select the population that we're going to enroll in these studies? You know, can we identify the highest risk people to participate in these studies so we don't need large N's? And, you know, how do we select the products? Are animals a gatekeeper? Do all the products as they move forward have to show efficacy in some kind of animal model? If so, which model? Is it the non-human primate model? Which non-human primate model? Is it a humanized mouse model? So these are all very difficult questions, and I think important questions that the field really needs to intensify their studies on, and in addition probably needs to do a much better job of phase 1-2 trials. So intensive PK, pharmacokinetic, pharmacodynamic studies, looking at how well a drug or an antibody or a monoclonal antibody that could be delivered systemically or even topically, and people are working on that. You know, how much drug gets into the genital tract if that's where infection is occurring? How active is it? Is it active when there's genital tract secretions around? Is it active when a woman's on hormonal contraception? Is it active after sex? All these kinds of questions, which actually can be addressed with fairly inexpensive, intensive studies.
And I guess another issue there is some of these drugs actually have higher concentrations in the rectum than they do in the genital mucosa. So you need to consider that as well in terms of your target population. Is that correct?
Absolutely, and that's also, I would defer, I think that's true for vaccines. Responses in men and women to vaccines we know historically are not identical.
Well, and in different populations as well, depending on what their history of concurrent infections is. But I think you're getting at a point that really is critical for our way forward in this field. And, you know, at the risk of stating the obvious, you know, for antiretrovirals, we have had now for decades a great biomarker, which is the viral load, and that allows us to make these early decisions and really put our resources behind the things we think are the very best because we have something we believe in. We don't have that for vaccines. And I think the point that the listener and the questioner raised is actually important for vaccines because, as you point out, it will get much more expensive to test vaccines because now your control group is going to have to be bigger. You're going to have fewer cases. What we really need for the vaccine group is the equivalent of the viral load for vaccines. And I think if we can do that, and broadly neutralizing antibodies potentially could do that, but the T-cell vaccines will be different.
Well,
then
if
I think
broadly neutralizing antibodies take 2 years to develop, that's not much of an early biomarker, right?
Well, I would go back to Julie's comment and just withhold judgment on that. I think that the 2 years is a number that you see in people who are already infected, who have damaged immune systems. If we're going to make a good vaccine, we're going to have to make it faster. But either way, I think that we need that biomarker for vaccines. And I do think if we don't have it, as these newer therapies come in, combination, PrEP or test and treat, we will have trouble getting out of the box in terms of testing new products.
So moving from two areas where we have pretty clear evidence of efficacy with some of the things that are available now, both test and treat and PrEP, let's talk a little bit about vaccines and where we are now with vaccines and what is the most exciting new science around vaccines?
Well, we've talked
touched
about
on it a little bit.
the promise of envelope immunogens potentially being able to induce neutralizing antibodies that recognize circulating strains broadly. I think, and then you alluded to the other one, Bruce, is recent exciting data in the non-human primate models looking at rhesus CMV vectors, which are designed, at least the constructs thus far, are designed to elicit T-cell responses. And in particular, they induce effector T-cell responses, both CD4 and CD8 responses. Some of the CD8 responses are quite unusual in that they not only are recognizing epitopes through MHC class 1 restriction, but also MHC class 2 restriction. And these are all findings that have been reported in several papers recently in Nature, earlier in Nature Medicine by the Picker Lab and Scott Hansen. These are very important because, first of all, it sort of redeems a little bit the T-cell concept in vaccines, so really sort of giving us a balance into the types of effector responses that could be protective with a vaccine. But secondly, the interesting things about these, that if they can be made safe in the human CMV component, then it's going to be important to see whether or not these could not only be useful for acquisition against HIV, but also as therapeutics. And in that case, they could potentially be important in certain cases in maybe trying to eradicate the virus.
Gary, in terms of the most promising science that we've got right now, do you have anything else to add to what's been said thus far and what Julie said?
You know, to me, Bruce, what's been really exciting to see has been our ability to dive into the immune repertoire and see exactly how we can generate these different types of broadly neutralizing antibodies. So we know from the work of a number of labs, Dennis Burton, Ian Wilson, Peter Kwong, John Muscola, Michelle Nussenzweig, and others, that there are 4 classes of broadly neutralizing antibodies. They're the ones against the CD4 binding site. You have the V1, V2 region. You've got some V3 glycan-directed antibodies, and then finally the membrane proximal region. For each of those, because we now are able to do deep sequencing, we can go into people who generate those responses. We can generate millions of DNA sequences from their B cells, and we can recreate the history of their immune responses. And then that allows us to sort of find a critical path that their immune systems took to generate those. That's really unprecedented. I mean, to have that level of understanding, it isn't just that it's analytical, but the nodes in that path become tools for us, because we can say, well, I've got to hit that node, so therefore I have to design a vaccine that will pull the immune system into that direction. So it's not only that we understand the structures, that we understand the antibodies. We're now beginning to acquire a fundamental knowledge of how to get there from a germline B cell, which is really the scientific problem for a vaccine. So it's exciting, it's unprecedented. It will still take time. These are really complicated problems. The one thing we don't understand, I think one of the critical scientific questions is in this whole process is what are the rules for clonal deletion? Why does a particular B cell get deleted?
Yeah, and so now you're talking about very fundamental immunology that actually applies beyond the HIV field. And I think to me that's one of the exciting things about all of the efforts that are going on right now, is that we're gaining insights from studying HIV that are going to be broadly applicable to understanding how the immune system deals with viruses and cancers in general. If we think about PrEP and vaccines side by side, probably Mike, the best efficacy from a PrEP intervention would be 96% protection with treatment of discordant couples.
Well, I think that you're mixing 2 studies, but it would be good. That'd be great if it was. So I think if you use an antiretroviral, I think if you treat the index case successfully, you should anticipate no transmission.
Right.
I think if a person takes a PrEP agent that works and takes it faithfully, one should accept no transmission. That would be, I think, the anticipated benefit.
So we have that, and then we have best vaccine efficacy that we have thus far is a questionable 31%. There's a question that's come in from Tim Crawford at the University of Colorado and C.G. Park from Korea, asking a pretty straightforward question. What is it that we actually want from a vaccine, and when are we going to call that a success? And we should probably hear about that from all parties.
Well, I don't think we know what we want. I mean, I think we've got good ideas. We've got carefully
Well, I guess there's two ways
conducted
to think
immune
about
correlate
it.
study with RV144.
Well,
that-
you know, it's either there are two parts of the question, or two ways to interpret it. What do we want? What kind of efficacy do we
Right.
want? And do we want protection from infection versus long-term control, sort of converting people into elite controllers?
Okay.
You know.
Right. So I think just as sort of taking Mike's view on this, that we would love to get efficacy up to close to 100%, and we want it to last for a really long time. So that's the ideal.
Good answer.
Okay. We're not likely to get that. We won't. I think that it's important to make a major goal protection against acquisition. Obviously, among those who become infected, if their disease burden is less, that would be important to show. But with early treatment, it will become harder and harder, I think, to sort out what's a vaccine effect versus just what's getting treatment in at the very beginning. But with acquisition, I think some modeling of that's been done. We'd love to see something around 60% or better, and that's a target goal.
And you agree with that, Gary?
Yeah, I think to have a public health effect, you're going to need a vaccine that would, and so just to get back to the original part of your question, I think you clearly want a vaccine that blocks infection, one that controls viremia, at least for prevention, is probably not going to be the ideal. It might have
Is it a general non-starter in the field now? That whole concept of trying to
for prevention,
get a sort of
probably.
a functional cure with a prevention modality?
Unless you control it so well that you effectively have no viremia.
But it's an operational non-starter, I think, because of the ethics of the vaccine trials, because I think going forward, infected people, rather than be allowed to reach a set point, and they're going to be treated. I think you're going to roll people into treatment, and we're going to get to that in the fourth section, I believe, of treating people immediately. So even if the vaccine did have some benefit along those lines, I don't think you'd be able to see it, because we're going to treat people because we think that's in their best interest.
Well, let's take one more question related to vaccines, having to do with what should be the gatekeeper for taking products forward, and who should be making those decisions. It's similar to the questions we were asking about PrEP before.
So you're talking about a preventive vaccine?
I'm talking about a preventive vaccine now.
Well, I think ideally, I completely agree with Mike's comment that we just can't take everything forward into an efficacy trial. So what we need are criteria that allow us to, as best as possible, predict whether we're hitting biomarkers that we have some faith in. I do think that that this is one of the advantages of the broadly neutralizing antibody paradigm, is that we do have wonderful assays for detecting both the potency and the breadth of broadly neutralizing antibodies. And I think most of us would say that if we had any vaccine in a phase 1 study in humans that would elicit an antibody that would neutralize 60% of strains with a potency of 10 micrograms per mil, we'd be pretty enthusiastic about moving that farther down the food chain.
So, Gary, can I just interrupt you? So do you think that antibodies should be measured only in the systemic circulation, or do you think we should be measuring those antibodies in the mucosa?
Let me put it this way. If you can't find broad neutralization at some point in the serum, I don't think you're going to find it in the mucosa. I think once you have it, then for sure we want to know that it gets to those sites as well. The other point, though, gets to your point, Bruce, about Lewis Picker's model. And I think likewise there, we need to come up with some immune correlates of T-cell protection that will give us the same conviction to move forward. And that may have to come initially from non-human primate studies, but I think we'll be in the same box by the time we're done.
Well, and actually by your criteria then, Lewis's CMV HIV vaccine would not go forward if it's getting 50% efficacy in an animal model. He he seems to reliably get 50% with no impact on the other 50%. Does that mean he's not likely to reach the bar, and that shouldn't go forward?
I would not. So what I would say is
I'm going to look and see if Lewis is on listening to this.
Well,
Hello out there, Lewis, if you're there.
I'll channel my inner Lewis for you and say, one of the things that Lewis does is he's testing in one of the most aggressive SIV models that there is. So if you're hitting 50% protection in that strain, that's impressive. What might be nice would be then to test it with different strains.
Yeah.
Right.
And if we had a random set of SIVs and you could protect 50% of the different strains, I think that would be
And maybe with a low dose exposure as opposed to the high dose exposure.
Yeah.
But staying with this model of plausibility, I would think that the most important thing is explain what is present in the 50% that allows them to be protected.
Right.
That's the way you go forward. Otherwise, you're doing a big black box experiment. If you could say, if they get this immune response, they are going to be protected, that's the path to a vaccine.
Well, I mean, I'm a scientist and I agree. Understanding mechanism is good, but if you had something that was giving you 80 or 90% protection in a monkey model and you didn't exactly know why, would you oppose that going forward?
Well,
Please tell me no.
No, but I do think that's how you get on these roads, because we're scientists. As a scientist, you're really compelled to say, I've really got to understand it at some plausible level. Otherwise, you make the Homo sapien into a black box.
Yeah, but I think the other point about that is that studies that have been done in the non-human primates, when they get to humans and the studies are carried out and you get an outcome, we've often been surprised by what we've seen in people versus what we did see in the monkeys. So you can't completely use that model as a way to then predict whether or not something should move forward. So there has to be a really good scientific rationale. Maybe it's something different from what's been tried before, with a strong scientific rationale, or just a vast improvement on what's been done before.
Well,
but I
some would
don't think
say-
that you can just use completely the non-human primate as a gatekeeper for moving forward.
Well, some would say the non-human primate has not been that bad in terms of predicting some of the vaccine failures that have happened. What do you say to those people?
I think that that if you really look at the data carefully, there are parts of those studies that really have been predictive. But I think that's the part that really needs to be done. And the studies need to be well designed and something similar to what might be the types of designs that are used in the clinical trials. And then the types of immune responses that are examined need to be looked at similarly. I think where we get into trouble potentially are where we're comparing responses of a certain vector in a non-human primate and in a human, when there's different tropism for those vectors, or one is pathogenic in one model and not in the other in its natural form. So that's I think where you can get into some difficulties in making those comparisons.
Where I think we've gone wrong with the non-human primate in the past, and it's not just with HIV. If you look actually at the history of polio vaccine development, there were a number of vaccines that were taken into humans because they worked well in non-human primates in polio, and they just flamed out. And I think where we have to watch it is not as much with the non-human primate, because I think there are, as animal models go, it's actually one of the best animal models for a human disease.
I would agree.
But there are differences, and one of the differences that I think we need to model better is that this reliance on just using one challenge strain, because that just doesn't happen in people.
Should we be using a swarm then?
I think we should be definitely looking at swarms. We have to make sure that they're kind of representative of the kinds of swarms we see in people.
And are we kidding ourselves with this low-dose challenge model where it takes longer to get infected? Is that, Mike, you're...
Well, I think there's no right answer, except that the greatest risk of transmission occurs with the acute infection patient, which has a very high viral load. Some people think that the viral load is, after 80,000 copies, there's no more advantage, that 80,000 copies is a maximum. But it's still a lot of copies, and then the virus is unique. Petrus Hahn has recently been working on interferon susceptibility. There's kind of potentially signature sequences. It's a homogeneous swarm that goes to the next person. So the swarm idea is complicated because the most likely transmission event comes from a homogeneous inoculum in somebody recently infected. But I do think the low-dose challenge model, you're trading off something of what happens in nature.
We call that a low-dose challenge model. It's a lower dose than the initial single high dose. It's really not that low a dose, and I think there are some advantages to the multiple exposure model. You know, we recently completed a study looking at a tenofovir disoproxil fumarate, that's the prodrug of tenofovir, intravaginal ring in non-human primates, and it worked beautifully in the low, quote, low dose or a lower dose multi-challenge model. We then tried out a new model where we gave the non-human primates high dose depo-provera repeatedly every 3 weeks over a prolonged period, and again saw protection in that model. So I think there's a lot of nuances in the models that we can work on and try to find a better model. I think the idea of using a swarm of viruses is very important.
So just to keep us honest here, there's a question that came in that's probably worth bringing up at this point before we finish up with vaccines, and that is, how do we get this far into this conversation without ever mentioning condoms, from Richard Gordon at Wayne State University? Somebody
I think that's
want to say something about that?
I
Have we
think
given up on those?
No, not at all. I think that every
That's
trial.
a given.
No, every trial we do, you can't do a trial or kind of be in the prevention field without assuming you're going to do everything possible to prevent an acquisition event. And that includes talking about safer sex, worrying about STDs, trying to avoid riskier sex practices, and making condoms available, which have been widely available. So I think a lot of the reduced incidence on a planetary basis is ascribed to exactly what he's describing. Now, in the couple studies, when we started out those couple studies, in our group and others, we anticipated 12% transmission events within couples per year. That was what the literature said. But when we got into all the prevention activities possible, then what we saw was less than 2% transmission events in the control groups. And other groups, Connie Kellum's group and others, have exactly the same level. That's really quite important, because had we seen no change in the control group, it would have been like we weren't trying to prevent acquisition. So I think all studies like that have to deploy all these strategies and understand that the acquisition rate ought to be drastically reduced. As you model a vaccine study, if you're deploying all these tools, you would say we're not expecting nearly as high an acquisition rate. So I agree with that comment completely.
So let me ask one more question about in the vaccine realm before we go on to cure. It's another question from somebody that's listening in today, Will Fisher at the Los Alamos National Lab. Basically asks, how can the rate of vaccine concept trials be increased? Dr. Walker mentioned opportunity costs, which apply at every step of the vaccine development pipeline. How do we go from a 2-year development time from an idea to having something to be able to test in a phase 1 trial? And can that time be shortened? Because that would make a huge difference, probably.
I think it could be. I'd preface what I would say about this with the caveat that Mike raised. I think we have to be clear about where we want to go with an increased number of trials. I don't think we can afford to do a lot of phase 3 trials. It's just going to be too expensive. But that said, I think probably what Will is referring to is the fact that we can get a lot of information out of iterative phase 1 studies, particularly in the broadly neutralizing antibody area. And that's where I think we can start to look for new technologies in protein production. I think continuous sort of fixed volume production apparatus can allow you to actually make new proteins quickly without investing in large-scale development. That could be a tremendous advance forward. I think in some cases, certain technologies lend themselves better to higher throughput. DNA vaccines as one example. Streamlining the IRB process and having standard informed consents. So there are ways of doing that. I think we just need to decide what we want to test.
Do you think it's possible that we can make an entirely synthetic vaccine and forego all of the all of the cell culture issues that slow things down?
I think at this
They're
point that-
nanoparticles
No, I think it
and
might be hard.
TLR.
At this point, I think that would be hard.
Is it worth pursuing?
Well, maybe
Low priority?
as we get more defined epitopes, I think it becomes a possibility. And sure, we should be opportunistic if any of those occasions arise. But most of the epitopes that we've seen for broadly neutralizing antibody are complex enough, and the peptide sequences are long enough that I think we have to rely on some kind of a biologic cell-based production. But we, I think, can be quite flexible in looking at new substrates for production, and certainly there are ways to accelerate that.
So I might add on
Yeah.
to Gary's comment, just to emphasize that I still believe the major holdup is going to be production. I mean, that's what we're experiencing. We have some ideas of what needs to come forward. We know we are going to probably need to test a lot of these envelope immunogens in order to eventually get one that does give the desired response. And it's a slow process right now, and I think it really
how big do those
needs to speed
studies
up.
need to be? How many people do you need to test one of these immunogens in to decide go, no go?
Well,
Is this 10,
I mean,
20,
typically
50?
these are sort of designed as a 10 vaccine to placebo, or they can go up. If you're adding some new adjuvant to it, then you're going to need to add more people to look at safety. But typically these are safe, and so you can make those smaller, as long as the signal that you're trying to achieve, that immune response that you're hoping to get, you can see clear winners and losers in that way. But I think the other thing too, though, is just that some of the if we're talking about GP120 versus GP140, 145, or whatever, sort of the larger those gets, then potentially they may be a little harder to make. So that itself can also take time.
Other comments before we move on to start discussing cure? Good, because time is marching on. Well, let me start the cure discussion and this discussion about latency and what to do about latency with a question that came in from Andrew Ward at Scripps. Does the reemergence of virus in 2 bone marrow transplant patients change the prospects for cure? So there's been a lot of excitement about the Mississippi baby, the Berlin patient, and then these individuals that got bone marrow transplant. Some question about the Mississippi baby and because of gaps in data, but certainly evidence here that what we thought looked pretty promising may not be so much. What do people think about that?
What do you think?
What do I think?
Right, you're the Harvard guy.
Yeah, right. I think that I think cure is something that we still need to pursue, and I am not optimistic about the current agents that are there, and I'm personally worried about keeping our eye focused on vaccine development, because I think for the long term, if we're looking at this as a global problem, which it is, I don't really see a clear, direct pathway towards implementation of a cure strategy at this point. Now, that's not to say that we shouldn't try and do that, but it's a question about where do we prioritize the resources? What do you think?
It's meant to be provocative. I actually think, well, first of all, I think everyone here is enamored of vaccines, but I actually think that we'll make more progress more quickly in the cure field than in the vaccine field for the following reasons. I think that, much as Gary indicated, the monoclonal antibody idea is so important because we'll learn a lot just writ large. And I think that every 10 years, the treatment field changes dramatically. And I cannot but imagine that all the work in the cure field will change the treatment field fairly dramatically. Now, a real sterilizing immunity cure, that may be quite difficult. But cure work that changes treatment seems inevitable to me. Along those lines, I think the other huge advantage is we know what we're looking for. We have the latency problem, which is kind of one problem. We have the kind of fact we never really eradicate replication completely. There's an ongoing replication in the tissues. We have a compartment problem. We know we need to get our drugs into every compartment. And I think that the kind of work is nascent right now, but heavily it's invigorating a whole generation of investigators. There's nothing more vibrant than going to one of these cure meetings, which started out with just a few people, and now you see hundreds of people clamoring for registration. And that's because they can see the kind of importance and the kind of reality, the immediacy of the results. There's the surrogate markers we talked about. There's increasingly good surrogate markers. There's pathways that are being developed, the disrupt latency pathway and a combination immunological intervention pathway. So I find it compelling, personally, and I think that the investment should be very robust, personally.
Betsy.
I think I agree that it's exciting and compelling, and it brings new scientists into the area. I'm maybe a little more skeptical, and I think we, again, need to pay heed to experiences with other pathogens. And we all know of, take the herpes virus family, cytomegalovirus, EBV, herpes simplex virus. We certainly have gotten nowhere with a cure with those kinds of latent viruses. Even
Have we really tried with those?
well, the second comment was going to be how, the amount of research effort and dollars that have been spent in some of those viruses is clearly less. But yeah, there have been efforts in some of those viruses, particularly CMV. But you're right, maybe we haven't put the same effort in. And maybe if we do it for this virus, the lessons learned from HIV can then be applied to these other viruses. But viruses have been around a long time. Persistent and latent viruses have been around a long time. I'm not sure this is going to be so easy.
Gary's going to come to my defense. I'm counting on you, Gary.
No, actually, I will. I won't disappoint you.
Thank you. I knew it. I knew I could trust in my friend.
Well, I think if you take a step back, I think the recent cases aside, if you look at the approach we've taken to cure, it hasn't been terribly thoughtful up to this point in time. And I think that there are a number of things we can now do with a little bit more wisdom and perspective that we didn't do in the past. We still don't know the basic mechanisms of latency, what's really happening at the chromatin level, but we have tools to figure that out. We're just figuring out what the pathways are that induce cell death as opposed to chronic infection. That's critical to understand at a molecular level and target it. If you look at the approaches that we use to try to cure, we use antiretroviral drugs. The antiretroviral drugs kill replicating virus. They don't actually kill a cell that's latently infected with virus. That's a different thing, killing the cell versus killing the virus. But yet we have all the tools to address that. And I think on top of that, we now, with the monoclonal antibodies and with our ability to engineer those, we can make antibody drug conjugates, we can make bispecific antibodies. These are more tools to try to get in and specifically deliver to those sites. The thing that I think we need most to really make a quantum jump in that field is we need the biomarker. Like, we need the equivalent of the viral load for latency. And while a lot of folks are working very hard and they're making good progress, I don't think we have it yet. But on the other hand, you have some commercial companies, places like Illumina, for example, that can do amazing things that allow us to understand at a molecular level, how much of a particular gene is present, whether it's methylated or not. I think we need to start engaging people with complementary interests, take a fresh look, and I think really push on this problem.
And for the cure AIDS field, we have the mouse model. We haven't mentioned the mouse model once. That's a big advantage. Now we have it's not cheap, and it's not that easy, but it's really much quicker. And those mice, I think, will make a contribution, in my opinion.
Yeah.
And I'm quite sure you have a cure AIDS agenda going on, Bruce. I'd like to hear what you're doing.
Well, we do actually, but I do have an ongoing bet with Steve Deeks. He claims that there will be a a cure long before there's a vaccine. And I'm on the vaccine side of that bet. What do you think my chances are? Not good.
Depends on what you call a cure and what you call a vaccine.
Okay.
You'll both win.
Oh, great. I hope Steve's listening. I think one of the exciting things about the cure field is that it actually brings the vaccine field sort of back together with the treatment field, and I wonder if anybody wants to comment about that.
Well, it gets back to what we were saying just a few minutes ago. I think that, obviously if we can, and that's another example where I don't think we've been necessarily so intelligent about how we've approached therapeutic vaccination, because what we've tended to do with vaccines is to say, in a person infected, we'll give them a vaccine like we would an uninfected person and expect it to do something that the natural infection already hasn't done. So we've got to start thinking about the vaccine, or a therapeutic vaccine, as only one of the cornerstones. We've got to add, some immunotherapeutic to it to complement what's missing from the immune system. So I think maybe thinking about it as more of an immunotherapeutic as opposed to a therapeutic vaccine. But clearly, if we're going to hold that virus in check, we've got to exert pressure on it any way we can, and the immune system is one of the most powerful ways to do that.
And there's a lot of movement in trying to find people with acute infection and treat them immediately, rendering their immune systems intact, rendering one virus as the enemy, and then identifying a strategy to provide an immunotherapeutic that, when you stop therapy, has the ability to constrain HIV. And this is, by the way, your elite controller career. I just summarized it.
Thank you.
If you'd like to talk about it.
Well, go ahead, Julie.
But I mean, I think that as yet we don't really know whether getting in, actively immunizing in people at risk for HIV infection, really how well we can do across the board once they become infected. We know that for vaccines that really haven't reduced viral load post-infection, we still have been able to see through the CIF analysis that there has been some pressure that's been put on the virus in terms of which viruses did get transmitted. And so I think we still have some room there to be able to act before infection occurs, and then potentially be able to get in and treat as early as we can. So that close follow-up and sort of moving from the pre-infection to acute infection in a seamless way to get at cure is going to be important.
Okay. Before we move on to closing remarks, maybe I can have you address another question that's come in, which basically is, as a junior person entering the field, where should they be focused?
Where their passion lies.
Good answer.
I'd ask Gary that as a junior person. I'd give Gary's advice.
Well, no. I think that Betsy's right. I think there are multiple areas in the field now that are scientifically very exciting. We talked about the B-cell ontogeny and structural biology. We've talked about latency. We've talked about, even downstream clinical trials, and we've talked about, antibody. There's a whole spectrum where we're talking about gene delivery of antibodies. There's a whole spectrum of activities that I think are really interesting. They're scientifically challenging. They're rigorous. They're going to open new frontiers. And I completely agree with you. People do best when they study the things that really excite them. And there's a large menu to choose from. So I'd just be open-minded and look at those alternatives and go for one.
And I'd throw in a couple of more. The action for HIV, where exposure occurs, is in the mucosa, and so we really need to know more about mucosal immunity. And so it's an incredibly challenging science, particularly in humans. But I think that more young brains really interested in this field could make a big difference.
Absolutely.
And do you think that along those same lines, understanding what's going on in tissues as opposed to peripheral blood is something that needs more attention?
Exactly.
Yeah.
And even optimization of some of the animal models like we talked about earlier.
Yeah. Well, I think we're almost out of time. Maybe we can just quickly go through with some last comment from everybody in terms of that initial question. Where do we put our priorities right now in balancing all this? Or is that a fair question? We didn't really answer it in terms of anything too specific.
Well, from my perspective, I probably no reason to repeat the things we've already discussed, but I do think that we need to make sure that we have a balance in what we do, a balance that maintains the effort on the basic science questions, the immunology, the virology, the infectious disease pathophysiology. And then I think, as Mike and Betsy and Julie have all pointed out, and you too, Bruce, the implementation. I think we're now all realizing that implementation is a science, and there's a continuum of when you investigate and when you apply. And my only kind of parting comment on this is one that we had discussed earlier, which is that think back to the smallpox vaccine. If we waited to understand the best way to implement the smallpox vaccine, we would have never eradicated smallpox. I think that one of the biggest gaps in our ability to wind down this epidemic is that I do think that public health departments and governments have to get started on some of these implementation measures, even if we don't understand how to do them perfectly. Yeah. There's a menu of them. I think we've heard them around the table. We should use the ones that are working and move forward. And where they're not working, we should use our science to generate new ones that will move them into place. And I think as long as we do that and build our foundation and advance into the application and the implementation, we'll be fine.
Julie, some closing comment?
Yeah, well, I agree with what Gary's saying. Some additional comments would be, I think we should continue to keep an open mind for what's out there, to really take some risks, to try some new things. And I think with that, apropos to your previous question, is to try to engage young minds to help us think of what those risks should be, and how to approach the problems in sort of their generation level of science. I think it's going to be important to do. And then finally, when we get surprises, just to be ready to adapt to those surprises very quickly.
Betsy?
I think we just add two other things to what's already been said. I mean, this is the classic example of bench to bedside and back again. And I think we need to be very careful. We still need a lot of the science, but we also need to be careful that as we do the clinical trials, we're collecting the right biological samples. So whatever the clinical trial outcomes, we can go back to the bench and figure out what went right and what went wrong. And at the same time, we need to do the implementation science in the way that Gary pointed out, where we're not sitting around, thinking about it. We're moving things forward rapidly, but do implementation science in a way that we actually learn from the implementation. So when oral PrEP is offered and people aren't taking it, we need to understand why, and we need to do really good science in understanding why, so that we can really advance protection.
Mike, some final pearls?
Right. Well, Gary's inspired me to be very historical, because I do think this is really quite a critical time, perhaps the most exciting time in HIV field in certainly in maybe 15 years. But we need to learn from history and avoid kind of hyperbole. We had treatment for syphilis in the 40s. We had treatment for tuberculosis for the last 25 years. We haven't treated our way out of these epidemics. And the mistakes that were made there were kind of hyperbole, like people losing interest once treatment is available, kind of resting on your laurels. This is the time to redouble all of our investments. That's probably the most important thing, is to maximize the kind of activities that are possible now that have been emphasized so much in our discussions, and then really seek the funding to redouble the investments at this moment in time because of our success and the excitement of the field. The other point as you do that is to know what not to do. The discipline that the pharmaceutical industry has left and right, they're able to kill agents pretty easily. They say, this isn't going to fly, we're done. We have a lot more trouble in the kind of academic industry. We need to ask the right questions, and I think that'll get us really far.
Thank you. Well, we hope you've enjoyed the event today, and we invite you to continue the discussion both online through the chat room and at the Keystone Symposia with these HIV conferences that are going to be occurring in March in Banff. There'll be a workshop at that conference specifically focused on this same topic and addressing questions that we didn't have time to discuss today. Because this is Keystone Symposia's first webcast, it's also very important to us and to them that we get some feedback. So if you could all please click on your survey button in your browser to answer a few brief questions, it would be greatly appreciated. And if you'd like to view this again or recommend it to someone you know, an archived version will be available next week. I'd like to say thank you to all the panelists, Mike, Betsy, Julie, Gary, and thank you to all those who participated online. Good afternoon.